Okay, so continuing on with the Chem 112 Organic Chemistry lecture series, we're going to be discussing Chapter 6 now, which is an overview of organic reactions, focusing on organic mechanisms and focusing on understanding organic reaction mechanisms and reaction diagrams. So, first and foremost, let's talk a little bit about this concept of polarizability, and let's talk about the standard definitions of nucleophiles and electrophiles. So, polarizability is the tendency to undergo a change in electron distribution in a bond or molecule. Generally, bonds are polarized based on the electronegativity of each bonded atom. So, for example, this carbon-sulfur bond is polarized in which sulfur has the majority of the electron density. So sulfur is electron rich. That's why it has a partial negative charge. While carbon is electron deficient, and that's why carbon has, in this case, a partial positive charge. It's really important to be able to look at a molecule and estimate how the bonds are polarized, as it can help you identify electron-rich and electron-deficient atoms. Okay, let's now talk about electrophiles and nucleophiles. So polar reactions occur between electron-rich and electron-deficient atoms. The electrophile is defined as an electron-poor species. A Lewis acid is an example of an electrophile. So for example, if we look at a carbonyl, this carbonyl is polarized. This carbon-oxygen bond is polarized. The carbon is electron deficient, electrophilic. The oxygen is electron rich. By the same token, if you have a carbon-chlorine bond, this chlorine atom is quite electronegative, and this carbon-chlorine bond is polarized such that carbon is electron deficient, electrophilic, while the chlorine atom is electron rich. There are a range of electrophiles, and electrophiles generally react with nucleophiles in polar reactions, and electrophiles will accept electron density lone pairs. So electrophiles are electron deficient species. Nucleophiles, on the other hand, a nucleophile is an electron-rich species. A Lewis base is an example of a nucleophile. One example of this would be an enolate, where we have this electron-rich oxygen. And as we can see by resonance, this carbon atom is also electron-rich, also nucleophilic. Another example of a nucleophile that we'll see in many common reactions in future chapters are pi bonds. Pi bonds can be nucleophilic, especially if they are electron-rich pi bonds. Other examples include anions, be it halide anions, carbon-based anions. Another class of nucleophile we'll see a lot, mostly in 113, are organometallic species such as Grignard reagents. The main point is that nucleophiles donate electron density from their lone pair or pi bond. So electrophiles are electron deficient, nucleophiles are electron rich. Now, within a molecule, there can be both electrophilic and nucleophilic sites. So molecules can have both electrophilic and nucleophilic sites of reactivity. Electrophilic sites are electron deficient, while nucleophilic sites are electron rich. To identify electrophilic and nucleophilic sites, it's important to take a look at resonance and inductive effects. For resonance contributions, you'll identify atoms with partial charge in one or more of the resonance structures. Oftentimes, the resonance structure that gives the most information in terms of electrophilic and nucleophilic character is the second best resonance structure. So in this case, for a carbonyl, for example, we can draw a resonance structure where we kick this pi bond set of electrons up to oxygen, we see that the oxygen is electron rich, it's nucleophilic. The carbon is electron deficient. The carbon is electrophilic. By the same token, this specific structure, this is known as an enamine, which has a lot of similarities in terms of reactivity with the enolate that we saw earlier on in this lecture. So an enamine, even though it's electron rich at nitrogen, it's also quite electron rich at carbon. And in fact, many enamines will react as nucleophiles, with the nucleophilic atom being this carbon atom here. So resonance structures can provide you a lot of information on what part of a given molecule is electron rich and electron deficient, which part of the molecule is nucleophilic versus electrophilic. Now, you can also consider inductive effects. Atoms bonded to more electronegative atoms are electron deficient, while atoms bonded to less electronegative atoms are electron rich. If we think about the carbon-boron bond, carbon is more electronegative than boron, so carbon is electron rich when it's attached to boron. It's much more electron rich, much more nucleophilic than usual. By the same token, if we compare the carbon-chlorine bond, chlorine is more electronegative than carbon, so this carbon would be electron deficient, electrophilic. Same for this carbon-sulfur bond. You can also take a look at some pretty interesting cases, looking at this nitrogen-bromine bond for a moment. This is an analog of succinimide, which is a reagent that we'll see later on. If we take a look at this nitrogen-bromine bond, because nitrogen is relatively electronegative, and you have this pretty massive inductive effect from these adjacent carbonyl groups. This pulls electron density away from this bromine atom, so this bromine atom is much more electrophilic than usual. And as we'll see later on, N-bromosuccinimide and related reagents are often used as an equivalent of a Br plus cation. So now that we've seen two ways to identify electrophilic and nucleophilic parts of our molecule, let's practice and let's indicate the atoms that are electron deficient and electron rich in the following structures. I'll do the first one, you'll do the next one. For this first case, we have a carbon-phosphorus bond. If you look up electronegativity values, carbon is actually more electronegative than phosphorus, so you can draw a reasonable resonance structure that looks like so. This is known as an ylide, and this is a general class of reagent known as a Wittig reagent. These are used for carbon bond forming reactions, specifically to form carbon-carbon double bonds. And the way that these Wittig reagents react is that the carbon atom in the Wittig reagent is the nucleophile. We can intuit that pattern of reactivity by looking at our molecule, thinking about electronegativity trends, and drawing some resonance structures. So in this case, the carbon is electron rich, the phosphorus is electron deficient. Carbon is nucleophilic, the phosphorus is electrophilic. So I'd like you now to work on these next two examples. So pause the video at this time, work through these next two examples, and then we'll discuss. Okay, so first and foremost, we can draw a contributing resonance structure where we kick the pi bond up. This generates a minor resonance structure, but nonetheless it has instructive value, because as we can see, this carbon has some electrophilic character. By the same token, if we kick the lone pair down, we can generate another resonance structure. This is mostly for completeness. As we can see, looking at the resonance structures drawn, these two oxygen atoms in this acetate anion are electron rich, while the carbon atom attached to the carbonyl carbon is electron deficient. By the same token, if we look at this conjugated ketone or this vinyl ketone, we can draw a resonance structure where we kick the pi bond up to the more electronegative atom, which is oxygen. This gives us the following resonance structure. And we can draw one more resonance structure where we move over our pi bond, and that gives us the following resonance structure. What this tells us is that these two carbon atoms are electron deficient, while the oxygen atom is electron rich. Okay, so we're able to take a look at molecules and identify which parts of the molecule are electrophilic, which parts are nucleophilic, and we can identify electrophiles and nucleophiles in a general way. Let's now talk a little bit about heterolytic bond cleavage and polar reactions. So, as we've seen in prior chapters, such as the acid-base chemistry chapter, we indicate the movement of electrons in reaction mechanisms using curved arrow notation. In heterolytic bond cleavage, the electrons shared between two atoms in a chemical bond are distributed to one atom during bond breaking. The curved arrow for bond breaking is drawn from the bond between the two atoms to the atom receiving the lone pair or electron density. So to describe the movement of two electrons, we use a double-headed arrow. It's important to be really careful with your arrow notation because the double-headed arrow is specifically referring to two electron movements. So for example, if we're looking at the heterolytic bond cleavage of this carbon-bromine bond, we draw an arrow from the carbon-bromine bond to the more electronegative atom, which is the bromine atom. Why do we break towards bromine? Well, it's more electronegative, and it can more readily accommodate a negative charge, while carbon, being less electronegative, can more readily accommodate the positive charge. So let's practice this. Let's look at the heterolytic bond cleavage for this carbon-oxygen bond. So just like before, we draw an arrow from the bond we're breaking to the more electronegative atom, and we make sure it's a double-headed arrow. This in turn yields a carbocation as well as water. One term that we're going to introduce a lot later on is the group that leaves, such as this bromine atom and water. We call this the leaving group. You'll see this term brought up multiple times in later chapters. Now, we call these types of reactions polar reactions as they involve heterolytic and unsymmetrical bond forming and bond breaking events. A classic example would be an acid-base reaction. We've seen this before. Here in this case, the nitrogen pulls off a proton from this acid, and in turn, we form a new nitrogen-hydrogen bond, and we break the oxygen-hydrogen bond. So because we're so used to drawing acid-base reactions, let's just practice drawing this acid-base reaction mechanism. So here we have an alcohol, and here we have a vinyl anion. So in this case, we draw an arrow from our lone pair to the acidic proton, and then we break the bond between the oxygen and hydrogen atom to put a lone pair on oxygen. Now, in terms of polar bond formation, in this case, two bonding electrons are donated by one reactant. To draw the mechanism for polar bond formation, the curved arrow for bond formation is drawn from the lone pair of one atom, or the source of electron density, such as a pi bond. So you can either draw it from the lone pair or a pi bond to the second atom that is part of the new bond. For example, in this case, if we wanted to form a bond between these two carbon atoms, we draw an arrow from our lone pair to the electrophilic atom, which is this carbon with a positive charge. This yields a new carbon-carbon bond, and this really completes the mechanism for this polar bond formation. So let's practice now. You have a carbocation, you have a bromide anion. Let's show how we can form a carbon-bromine bond. So in this case, we have our lone pair. We draw an arrow from the lone pair to the other atom participating in the bond, which is typically going to be the atom that's electron deficient, in this case, our carbocation. And from here, that forms the desired carbon-bromine bond at the cost of one of our lone pairs on bromine. Okay, let's now practice drawing some polar reaction mechanisms. And I'd like you to draw arrows to complete this two-step mechanism. And I'd like you to tell me what is the byproduct in this first step. So let's take a look at what's happening here. So here we have a carbon-iodine bond. We break that bond to make a carbocation, and then we form a carbon-bromine bond. Okay, so we've seen the steps to draw bond formation and bond breaking. So I'd like you to pause the video at this time and practice and try to draw out arrows to complete this two-step mechanism. Okay, so first and foremost, we need to break this carbon-iodine bond. To do this, we draw an arrow from the carbon-iodine bond. And again, we're drawing an arrow from the given bond to the most electronegative atom. So in this case, we draw an arrow from the carbon-iodine bond to iodine. So as a byproduct, we would generate the iodide anion along with our carbocation. For the second step, we're forming the carbon-bromine bond. So we draw an arrow from the bromine atom lone pair to our carbocation. This forms the carbon-bromine bond. For those curious, and for those who are reading ahead, you've actually drawn a full mechanism for what's known as the SN1 reaction. So as you can see, by understanding and studying the key steps in polar reactions, we can put those steps together to draw the mechanisms for more complicated reactions. So because when we look at polar reactions, we often generate cations and anions, we have to now talk about and review some anion stability trends. Polar reactions involve anions as reactants or products, so anion stability must be considered. In terms of factors influencing anion stability, atom electronegativity and size plays a huge role. Anions are more stable on large electronegative atoms. Additionally, in terms of resonance effects, anions stabilized by resonance are more stable. Aromatic anions are very stable. For example, looking at this cyclopentadienyl anion, we have 6 pi electrons, which follows the 4n plus 2 rule. So this is an aromatic anion. Aromatic anions are especially stable, and it's important to be able to recognize aromatic anions when they come up, as generating an aromatic species can often be a driving force that pushes reactions to completion. Another factor that influences anion stability is induction. Electronegative groups can stabilize adjacent anions. So, for example, we can see the effect in terms of inductive and resonance effects by looking at pKa values. So, for example, as we've seen, if we compare acetic acid to chloroacetic acid, the more withdrawing groups that we place, that in turn stabilizes the anion, and thus we see that by the fact that the parent acid is more acidic. Another common group that is often a textbook electron withdrawing group is the NO2 group, which is the nitro group. The nitro group is strongly electron withdrawing, not only by induction, but it's also a resonance acceptor. So nitro groups in particular are very good at stabilizing adjacent anions. Just to show that, if we had an anion adjacent to this nitro group, it wouldn't only be stabilized by induction, but you can also draw a resonance structure where this anion is delocalized into the nitro group. So electron withdrawing groups can stabilize anions both inductively and via resonance. Another effect to consider are orbital effects. Anions are more stable if the lone pair electrons are in a lower energy orbital, where the sp orbital is the lowest energy orbital and gives the most stable anion, while the sp3 orbital is the highest energy orbital, which yields the least stable anions. In terms of solvation, this is more of a minor effect. The less steric congestion around the anion, the more stable the anion. So for example, if you compare a secondary to a tertiary alcohol, you'll notice that the tertiary alcohol is the least acidic. The reason being is that if you generate this tertiary alcohol anion, the bulky tertiary alcohol blocks solvent and limits the ability of solvent to interact with and stabilize your anion. So this is more of a solvation effect. Now, we've talked about anions. Let's talk a little bit about cation stability. Polar reactions can involve cations as reactants or products. Cation stability is key. Carbocations, which is a carbon-centered cation, are electron-deficient species. Carbocations are stabilized by adjacent bonded atoms. This is why, if you think about carbocation stability, primary carbocations are the least stable, while tertiary carbocations are the most stable. So for example, if we had a choice between these two carbocations here, with this one being a secondary and this one being a tertiary carbocation, we would say that this tertiary carbocation is more stable. It's important to be able to identify the most stable carbocation, as in many reaction mechanisms, you will have a choice on what type of carbocation to form, and reactions will prefer, in most cases, to generate the most stable, which is the most substituted carbocation. To answer the question, why are carbocations stabilized by substitution, we have to bring up this idea of hyperconjugation. Hyperconjugation stabilizes carbocations, and this effect increases with adjacent groups. In hyperconjugation, you have the overlap of the empty carbocation p orbital with the adjacent sigma bonding molecular orbital. So we have the carbon-hydrogen sigma bond, and it overlaps with the carbocation p orbital, and this in turn stabilizes the carbocation by donating electron density. The more groups you have adjacent to your carbocation, the greater this hyperconjugation effect. So for example, if you have an adjacent methyl group, the three carbon-hydrogen sigma bonds can be involved in hyperconjugation, and thus we're essentially spreading this positive charge over more atoms. The more we can delocalize that carbocation positive charge via hyperconjugation, the more we can stabilize your carbocation positive charge. One thing I do want to be clear, this is not as strong as resonance delocalization. This is a weaker orbital overlap, but it is still a stabilizing interaction. The more groups you have adjacent to your carbocation, the more substituted your carbocation, the more stable your carbocation will be. Let's talk about some factors that can be problematic for carbocation stability. Let's talk about inductive effects. Carbocations are destabilized by electron withdrawing substituents that are not pi donors. Examples of electron withdrawing groups include the trifluorocarbon groups, trichlorocarbon groups, nitro groups, nitriles, carbonyls, carboxylic acids, sulfonyl groups, and nitro groups. The reason why the carbocation is destabilized is you already have an electron deficient carbon and you have a withdrawing group pulling electron density off that electron deficient carbon, and that is destabilizing. So if you have a choice between the two carbocations, the carbocation that has the carbocation adjacent to a nitro group is going to be less stable than just a normal secondary carbocation that's not adjacent to an electron withdrawing group. So we'd say this carbocation shown below would be the more stable carbocation. So keep an eye out for electron withdrawing groups. They're highly destabilizing for carbocations. Let's talk now about cation stability in the context of resonance effects. So carbocations are stabilized by adjacent pi systems as the cation can be delocalized via resonance. Resonance donor groups can stabilize carbocations by donating electrons into the carbocation p orbital. So for example, in terms of delocalization effects, if you have a carbocation adjacent to a pi system, we can delocalize that positive charge throughout the pi system. This is why, in general, carbocations adjacent to pi systems are more stable, and this effect is additive. For example, even though this carbocation looks like it's a primary carbocation, in reality, it's much more stable than a normal primary carbocation because it can be delocalized via resonance. And it can also be delocalized to the secondary position. Anytime you have a carbocation adjacent to an aromatic ring, we call this a benzylic carbocation. Benzylic carbocations are especially stable because they can be delocalized throughout this entire pi system. To really drive home the point that the more you can delocalize your carbocation, generally the more stable your carbocation will be. If we look at this bottom example, being able to spread this positive charge across 5 atoms in a pi system, this carbocation on the left will be more stable than the carbocation on the right, where we can only spread this positive charge over 3 atoms. So carbocations are stabilized via resonance. This is a very important idea. Now, in addition, carbocations are stabilized by adjacent resonance donor groups. Anytime you have an atom with a lone pair, it can stabilize your carbocation by donating electron density into the carbocation by forming a pi bond. So anytime you see a carbocation adjacent to an atom with a lone pair, even a halogen, in even a traditionally electron withdrawing in group by induction, that carbocation can be stabilized via resonance donation of a lone pair to form a pi bond. As you can see, by donating electron density and forming a pi bond, you can stabilize that carbocation by delocalizing that positive charge across multiple atoms. And I want to be clear, these resonance donor effects are much stronger than inductive effects. That's why, for example, a carbocation adjacent to a chlorine group is in fact more stable than just a normal carbocation without the adjacent chlorine group, because this chlorine is a pi donor. The last effect we have to talk about in terms of carbocation stability is this idea of orbital effects. Carbocations are electron deficient species, and they're destabilized the closer they are to the nucleus. Increasing s character results in a shorter orbital that is closer to the nucleus, which destabilizes the electron deficient carbocation. So the sp3 carbocation is going to be the most stable carbocation in general, while an sp carbocation is going to be the least stable. It is important to note that formation of an sp2 or sp carbocation is relatively rare. That's why, for example, if you were to compare an sp3, quote unquote, versus an sp2 derived carbocation, the sp3 derived carbocation will, in general, be more stable. Note that these terms are derived. Um, the reason why I use that phrasing is that this carbon here it's sp2, but it's a carbocation that's derived from an sp3 carbon. For example, if you take this carbon here, which is sp3, and it reacts to make a carbocation, you will get the following carbocation that is sp2 hybridized, and it's derived from an sp3 carbon. Now, to explain why the alkyl carbocation is generally more stable than the alkenyl and the alkynyl carbocation, you can use the orbital argument. It also has to do with, and it's very much focused on how this carbocation is handled. In the alkyl carbocation, you have an available and empty p orbital, and that available and empty p orbital can participate in hyperconjugation and help efficiently stabilize your carbocation. When you start to generate carbocations of the alkenyl and alkynyl variety, it's more difficult for you to stabilize the carbocation. And if you take a look at the hybridization of your carbon, the carbon is generally using orbitals with more s character, which are shorter, held closer to the nucleus, and in turn destabilize your carbocation. So in general, you're primarily going to see alkyl carbocations. Alkenyl and alkynyl carbocations are rare. So let's now practice what we've learned, and let's indicate the more stable carbocation in each of the following cases. So I'm going to do this first example. So in this case, we have a carbocation that is at the benzylic position. It's adjacent to an aromatic ring, and we can delocalize this carbocation throughout the aromatic ring. Here we have a carbocation adjacent to a pi bond. We call this an allylic carbocation. And in this case, we can only delocalize the carbocation across these three atoms. Because the carbocation on the left can be delocalized throughout the entire aromatic ring, the carbocation on the left will be more stable. Let's look at an example of atom effects. I'll do the first one, you'll do the second one. So here we have a choice between a secondary carbocation versus a secondary carbocation that's adjacent to an alcohol. As we discussed, atoms with lone pairs can serve as pi bond donors and in turn stabilize the carbocation. So the carbocation adjacent to the alcohol is going to be more stable. Let's take a look at this next example, and I'd want you to pause the video and tell me which of these two carbocations are going to be more stable. Okay, let's discuss. So here we have a secondary carbocation versus a tertiary carbocation. The more substituted tertiary carbocation is going to be more stable. Let's look at this next case. Let's look at this next example up here. Comparing these two carbocations, I'd like you to take a moment and then tell me which of these carbocations is going to be most stable. Okay, so this nitro group is electron withdrawing. So we want to place our carbocation away from this withdrawing group. So the carbocation on the right stays as far away as possible from this electron withdrawing nitro group. Thus, the carbocation on the right will be more stable. Okay, last case now. Let's take a look at these three carbocations, and I'd like you to tell me which of these carbocations is most stable. So you have an alkyl-derived carbocation, an alkenyl carbocation, and an alkynyl carbocation. In this case, it's going to be the alkyl-derived carbocation. This is due to orbital effects. The less S character, the more stable your carbocation. Or phrased another way, the less S character, the less unstable your carbocation is. Okay, these next two are special cases and require you to think a little bit about the geometry and structure of your carbocation. So I'm going to talk about this left example first, and I'll guide you through this left example because it requires you to pay attention beyond just what's drawn. So this carbon here, this carbocation here, this carbon, if you take a look at this carbon in the ring, we have to think a little bit about the geometry and the bond angles that this carbocation is demanding. So if we take a look at the hybridization of this carbon, we have 1, 2 attached groups and a pi bond. So this carbon is almost sp-like in terms of hybridization. So it demands that this bond angle is going to be about 180 degrees. Now, the ring cannot accommodate that large bond angle. If you try to make this carbocation with a model, you'll see that if you wanted to adopt the ideal bond angle, you'd end up almost breaking the ring with the amount of strain you're putting on it. So this carbocation is highly strained. So by virtue of this carbocation existing, it puts a lot of strain on the overall ring and the overall molecule, and thus it is less stable as a carbocation. I wanted to bring this point up because you need to be very careful whenever you have carbocations and other structures with defined geometries in a ring that the ring can accommodate the angle strain introduced by your carbocation. So the carbocation on the right is more stable just because it has more flexibility to accommodate an ideal bond angle. You don't have a ring to worry about for this linear carbocation. The example on the right, I would encourage you to remember that anti-aromatic systems have 4n pi electrons and they're very unstable. With that hint in mind, I'd like you to pause the video and I'd like you to tell me which of these two carbocations is more stable. So at first pass, you would look at the carbocation on the left and say, man, I can delocalize that carbocation across the entirety of the ring. I have 2 pi bonds adjacent. This is great. But if we count, we have 4 pi electrons. So this is an anti-aromatic system, which means it is very unstable. So the carbocation on the right is more stable because it is not an anti-aromatic system and doesn't have to pay this huge penalty associated with the fact that anti-aromatic systems are unstable. So just be on the lookout. It is very, very hard. Functionally, it never occurs. You're not going to see formation of anti-aromatic carbocations or anions for that matter. They're that unstable. We've talked about heterolytic bond cleavage in detail. Let's talk about homolytic bond cleavage and radical reactions. So in homolytic cleavage, you have a symmetrical bond breaking where electrons in the bond are evenly distributed to the two atoms as the bond is broken. To signify the movement of a single electron, half arrow notation is used. Looks something like this, almost like a fishhook arrow. When you're dealing with homolytic bond cleavage, you're going to make what are known as radicals, which are a species with an incomplete octet and an unpaired electron, which is represented by a single dot. To draw symmetrical bond breaking or radical formation, two half arrows are drawn from the bond to each atom participating in the bond. For example, if we are breaking the bromine-bromine bond in a homolytic manner, we draw a half arrow or a fishhook arrow from the bromine-bromine bond to each bromine atom, and that yields two bromine radicals. Let's practice this skill and let's show how we would break this oxygen-oxygen bond. We draw a fishhook arrow to each oxygen atom. So we took 2 electrons in our bond and put 1 electron on each of our oxygen atoms respectively. This yields an oxygen-centered radical, which as we'll see later on, this is a commonly drawn intermediate that kickstarts a lot of radical reactions we'll be talking about later on. If we wanted to show symmetrical bond formation, the half arrow is drawn from the bond or radical contributing the electron to the location of the new bond formed. So if we wanted to form this carbon-bromine bond from these two radicals, we draw a fishhook arrow from each of the radicals to the new bond that we would form, and that yields the new carbon-bromine bond. Let's practice this. So here we have 2 radicals. Let's show bond formation. I'm going to draw 1 fishhook arrow and the second fishhook arrow, and that in turn yields 2 electrons that we need for the carbon-bromine bond we want. Now, for those wondering, how do we know whether radical formation is feasible? We can look up tables of bond dissociation energy, which is the amount of energy required to break a given bond to produce 2 radical fragments when the molecule's in the gas phase at 25 degrees Celsius. Again, these are standard tables, so we have to have these rider stipulations. These bond dissociation energy tables are pretty useful for helping you get a sense of how easy it is to break a bond through radical processes. Let's talk now about some mechanistic features and drawing mechanisms for radical reactions. So radical reactions follow this idea of a radical cycle. Essentially, anytime you make a radical, you have to show what happens to that radical to eventually get a non-radical species and terminate the chain. So the first step in radical reactions is initiation, where two radical species are formed from homolytic bond cleavage. This can be done with heat or light. For example, breaking the bromine-bromine bond or breaking the oxygen-oxygen bond to generate a pair of radicals. The next step is propagation, where the radical reacts with another molecule to yield a new radical. Propagation can occur via two major ways: abstraction, where an atom is removed from a molecule to yield a new molecule and a new radical. Essentially, the radical goes and steals a hydrogen or other atom from another molecule. In this case, we form a new bond and we break a bond, and that in turn yields a new bond and a new radical. So for abstraction, you take one electron from the bond that you're breaking and place it on one of the atoms. The other electron goes to form the new bond. Propagation can also occur via addition, where the radical adds to an alkene to yield a new radical. So in this case, we take one electron from our radical, one electron from the pi bond, and the other electron gets used to form a new radical. So we've formed a new bond and we've generated a new radical. Propagation can repeat multiple times with different pairs of molecules, but eventually the radical chain undergoes termination, where two radicals react to generate a stable product. So you have your two radicals, they each throw in one electron apiece, and that gives your new bond. And as you can see, at the end of the termination step, we have no radicals remaining. Now, because we're talking about radical reactions, it's important to discuss radical stability. And radicals share a lot of similarities with carbocations in terms of stability trends. Radicals are electron deficient, and radicals are stabilized by adjacent bonded non-hydrogen atoms. So radicals are stabilized via substitution, with tertiary radicals being the most stable, while primary and methyl radicals are the least stable. So if we had a choice between this primary or tertiary radical, we would say the tertiary radical is more stable. In general, the most reactive carbon-hydrogen bond to abstraction is the carbon-hydrogen bond that would generate the most stable radical. Just like carbocations, radicals are also stabilized via resonance effects. Radicals are stabilized by adjacent pi systems via resonance. So, for example, an allylic radical is going to be more stable than even a tertiary radical. Resonance effects are generally stronger than substitution effects. So if you have a choice between this primary radical versus an allylic radical, the allylic radical is going to be much, much more stable. Now, because it's a radical, one thing I would like to show you is to show resonance delocalization, you are going to have to use the fishhook arrow notation. Okay, let's talk now about resonance, inductive, and orbital effects for radical stability. Resonance donors can stabilize radicals by donation of electron density into the radical p orbital. So because radicals are electron deficient species, they can be stabilized via pi donors, just like in any other case that we've seen. So, resonance donors can stabilize radicals by donation of electron density into the radical p orbital. So radical stability increases with adjacent lone pair donors. So for example, even though a halogen is traditionally thought of as electron withdrawing, because this halogen has an available lone pair, you can donate electron density into the electron deficient p orbital and stabilize your radical. So for example, if you have a tertiary radical versus a radical adjacent to an alcohol, the radical adjacent to the alcohol, which is a pi donor, will be more stable because that oxygen atom can donate electron density via resonance. By the same token, as radicals are electron deficient, radicals are destabilized by electron withdrawing groups. So for example, if you have a CF3 group, a nitro group, a carbonyl, a carboxylic acid, or a sulfonyl group, these electron withdrawing groups destabilize carbocations considerably. And they also destabilize electron deficient radicals considerably. So for example, if you compare a radical where we have a radical that's tertiary versus a radical adjacent to a sulfonyl group or a sulfonic acid group, the tertiary radical is going to be more stable because the radical adjacent to this sulfonyl or sulfonic acid group is destabilized by the electron withdrawing group. Radicals are electron deficient species. Finally, talking about orbital effects, increasing s character results in a shorter orbital that is closer to the nucleus, which destabilizes the electron deficient radical. That's why alkyl-derived radicals are more stable compared to radicals derived from alkenes and alkynes. This has to do with hybridization effects. This radical, which is sp3-like, that orbital has less s character, farther from the nucleus, more stable radical. The radical that is sp-like, held in an sp orbital, is going to be less stable because that orbital is held closer to the nucleus and is destabilized by nuclear charge. So if we have a choice between these two radicals, the alkyl radical is going to be more stable. So a lot of the stability trends for radicals mirror that of carbocations. Let's now talk about one last component of radical stability trends, which focus on atom effects. So as radicals are electron deficient species, radicals are destabilized when they're placed on an electronegative atom. Radical stability decreases left and to the right along the periodic table. So carbon-centered radicals are most stable, while halide-centered radicals are the least stable. Generally, radicals are also stable on larger atoms as the electron is placed farther from the nucleus and the electron density is distributed over a larger area. So considering the first stability trend, if we compare an oxygen versus a carbon-centered radical, the carbon-centered radical will be more stable as carbon is less electronegative, and in turn, that means the radical is going to be a little bit more stable. Going off of the size trend, as radicals are more stable when they're placed on larger atoms, for example, the radical on the larger iodine atom is going to be more stable than a radical on the smaller fluorine atom. This also has to do with the fact that as you go down the periodic table and you look at larger atoms, they're also less electronegative. So if we compare a chlorine versus an iodine-centered radical, the iodine-centered radical will be more stable. Now, given all the radical stability trends that we've seen, let's identify the most stable radical in each set of molecules. I'd like you to pause the video at this time, work through these 4 examples, and then we'll discuss. Okay, so first and foremost, we have an allylic versus a benzylic radical. The benzylic radical can be delocalized across multiple atoms, and as a result, this benzylic radical is going to be more stable. Looking at this next case, we have a choice between oxygen versus nitrogen. In either case, we can stabilize this radical via donation of electron density. As we have seen, let's take a look at the resulting radical that we'd make. As we've seen, radicals centered on nitrogen are more stable. Nitrogen is also a weaker withdrawing group. It's less electronegative, so nitrogen is also a better donor. So you would expect that this radical on the right will be more stable than the radical on the left. In either case, both of these radicals are quite stable. Let's look at this next case. Here we have a tertiary versus a secondary radical. Tertiary radical is going to be more stable by far. Here we have inductive effects. So we have chlorine, which is a strong inductive withdrawing group, while bromine is a slightly weaker withdrawing group. As radicals are destabilized by withdrawing groups, the radical on the right is going to be more stable because it's adjacent to the weaker inductive withdrawing group. Okay, let's talk a little bit now about reaction enthalpy. So whenever we break chemical bonds, this costs energy, while forming chemical bonds releases energy. In combination, the act of breaking high energy bonds and forming low energy bonds releases energy. Exothermic reactions form stronger bonds in the product than in the reactant, and they release energy. Endothermic reactions break stronger bonds than they make, and they require energy. So we can talk about reactions overall by looking at reaction enthalpy and the heat of reaction. Exothermic reactions have a negative reaction enthalpy, while endothermic reactions have a positive reaction enthalpy. Exothermic reactions release energy, while endothermic reactions absorb energy. We can talk about some factors that influence reactions using this idea of enthalpy effects. Reactions can be driven to completion by a favorable reaction enthalpy. Some common features of reactions favored by enthalpy include, one example would be the replacement of weak pi bonds with strong sigma bonds. This is known as an addition reaction. Reactions that form strong sigma bonds are generally favored by enthalpy, as you can see by this enthalpy of reaction of negative 55 kilojoules, making this an exothermic process. Another factor that can often drive reactions to completion is the formation of strong sigma or pi bonds. Examples of this include silicon fluorine, silicon oxygen, and phosphorus oxygen bonds. These are sort of the textbook strong bonds that are formed as the thermodynamic sink in many reactions. Formation of these very strong sigma or pi bonds can be used to help drive reactions to completion. Classic example, the Wittig reaction forms this very strong phosphorus oxygen bond, and that makes this overall reaction process quite favorable. We've talked about enthalpy. Let's talk about entropy now. So entropy is a measure of the number of microscopic configurations and microstates a system can occupy. Essentially, entropy is a measure of the disorder in a system. A classic example of entropy that we see in the context of organic chemistry is in cyclic versus acyclic systems. Acyclic systems are flexible and have a greater number of accessible conformations than cyclic systems. In general, acyclic systems are higher in entropy. In terms of physical states, gases are generally higher in entropy than solids and liquids. They have more accessible possible configurations and microstates. Practically, how do we see entropy effects in the lab? How do we see these effects come up? So reactions can be favored by increasing entropy, increasing the net disorder or degrees of freedom accessible by a system. Common features of reactions favored by entropy include the generation of multiple products. For example, in elimination reactions, you start with one reactant and end up with 1, 2 products. This is favored by entropy. Another factor that is favored by entropy is the formation of gaseous products. For example, decarboxylation makes carbon dioxide, which is a gas, which is heavily favored via entropy. The last case that you often see of reactions where entropy helps in, where if we consider reaction entropy, the reaction is favored via entropy, would be ring opening. For example, ring opening via elimination. If you open the ring, you end up with an acyclic system, and thus the net reaction entropy is positive, and the reaction is often favored when we take entropy into consideration. Now, when we're juggling entropy and enthalpy effects, if we consider both enthalpy and entropy for a reaction, we get to this idea of Gibbs free energy. So the ratio of products to reactants is controlled by the Gibbs energy of reaction, called delta G, that represents the change in Gibbs free energy between the products and reactants. Delta G is defined as enthalpy minus T delta S. So we have both the enthalpy and the entropy terms accounted for. From now on, we're primarily going to be focusing our discussion on Gibbs free energy. We can classify reactions as either exergonic, with a Gibbs free energy value of less than 0. These are product favored reactions. Or endergonic reactions with a Gibbs free energy value of greater than 0. Now, as you've noticed by this equation for Gibbs free energy is temperature dependent. As the temperature increases, the entropy term becomes larger. At high temperatures, reactions that have positive entropy values that increase entropy are more favored. As a corollary, at high temperatures, reactions that have negative entropy values that decrease entropy become less favored. So part of the reason why we have to and why we discussed enthalpy and entropy effects is because Gibbs free energy is temperature dependent. Depending on the temperature, the entropy term will have a larger or smaller effect on the Gibbs free energy value. Now, as mentioned, Gibbs free energy can be related to the ratio of reactants and products. The Gibbs energy of reaction is related to the reaction equilibrium constant through the following equation, which you've probably seen in Chem 102, where delta G is related to R, which is a constant, the temperature, and the natural log of the equilibrium constant, where the equilibrium constant is the ratio of product and reactant concentration, or activities, if you've read far enough along into the 102 curriculum to begin the discussion of activities. The equilibrium constant is the ratio of product and reactant concentrations at equilibrium. For reactions that are product favored, these are exergonic reactions with a Gibbs free energy value less than 0 and an equilibrium constant greater than 1. For reactions that are reactant favored, one moment. So for reactions that are product favored, these are exergonic reactions with a Gibbs free energy value less than 0 and equilibrium constant greater than 1. For reactions that are reactant favored, these are endergonic reactions with a Gibbs free energy value greater than 0 and a K equilibrium value of less than 1. Now, just because your calculated Gibbs free energy of reaction is greater than or less than 0, it doesn't mean the reaction under every single set of conditions will be reactant or product favored. We have to talk about Le Chatelier's principle, which is the idea that when you set up or begin to run a reaction, you will generate and have reactants and products sitting in your reaction at a defined concentration. The reaction will proceed to drive the reaction back to equilibrium. The way that we exploit Le Chatelier's principle as organic chemists is by increasing the concentration of reactants, we can drive the reaction towards product favorability. The classic example is for hemiacetal and hemiketal formation, where we essentially use a large excess of reactants, and in turn, the K value for the actual reactant and product concentrations becomes much less than K equilibrium, and that in turn means using an excess of reactant drives the reaction towards product favorability. So whenever you see a reaction where the reaction conditions call for a huge excess of a reactant or reagent, sometimes that's because they're exploiting Le Chatelier's principle to push an otherwise reactant-favored reaction towards the products. This has been discussed extensively in 102. I just wanted to showcase one example of it in the context of organic chemistry. So why does this matter? Let's do a case study now, looking at the temperature effects on elimination versus addition reaction pathways. So as a general observation that's been found running a range of experiments, as the temperature of reaction increases, more elimination products are observed. Now, let's take a look at substitution versus elimination. In substitution, you just have replacement of your leaving group with your nucleophile. In elimination, you have elimination of your leaving group to generate the elimination product along with some additional products. Let's think now about how will the entropy term differ comparing substitution versus elimination. In substitution reactions, we have 1, 2 reactants, and we make 1, 2 products. In elimination, we have 1, 2 reactants, and we make 1, 2, 3 products. So in elimination, delta S is greater than 0 for elimination. We are increasing entropy. Now, why does that matter? As temperature increases, the entropy term plays a bigger role. So, as we increase the temperature, elimination becomes more favored. And it's because of this positive entropy term. One additional thing to note is that as the temperature increases, the Gibbs energy of activation for elimination also decreases, allowing this somewhat less favored pathway to predominate at higher temperatures. So this is a very tangible example of how enthalpy and entropy effects have and play a role in the outcome of reactions. As we can see quite clearly, because elimination has a positive entropy term, at higher temperatures, elimination becomes more and more favored to the point where it eventually becomes the predominant pathway. Let's talk now about reaction energy diagrams, specifically activation energy and transition states. The highest energy point in a reaction step is called the transition state for that step. A reaction can have multiple steps, so I just want to put that note. The energy needed to go from reactant to transition state is the activation energy, which is noted as the Gibbs energy of activation. The activation energy is different for the forward and reverse reactions, even though the forward and reverse reaction share the same transition state. So for example, here's the activation energy for the forward reaction. Up top is the transition state. And here is the activation energy for the reverse reaction. So this is an example step. This is an example diagram for what's known as electrophilic addition, which is a type of reaction. When we look at any reaction energy diagram, we look at each step of the reaction individually. Given what we've seen previously, is this step exergonic or endergonic? So in this case, if we look at the overall Gibbs energy, we see the overall Gibbs energy is greater than 0. So this would be an endergonic reaction step. You're going to want to really be comfortable identifying steps as exergonic versus endergonic. And the reason for that is it allows us to further describe and discuss the transition state and discuss how the reaction will behave and proceed. So let's now talk about activation energy and reaction rates. So the Arrhenius equation allows us to relate the Gibbs energy of activation and the reaction rate constant via the following equation. As you can see, again, there is some temperature dependence, a substantial amount of temperature dependence. In general, the reaction pathway with the smaller activation energy will occur faster and more product from that pathway will be preferentially generated. We call this the kinetic product pathway. Additionally, the rate limiting step is the slowest reaction step with the largest activation energy. The rate limiting step controls the product selectivity for a reaction. So, looking at this full energy diagram now, we have two steps. 1, 2. We see we have 1 Gibbs energy of activation for the first step and a Gibbs energy of activation for the second step. We also have the overall reaction Gibbs energy. Looking at this energy diagram now, I want you to tell me whether step 1 or 2 is rate limiting. What is the rate limiting step? What is the step with the largest activation energy and thus the slowest step? In this case, step 1, this first step is rate limiting. To answer the question, is the reaction overall exergonic or endergonic? We look at the overall reaction Gibbs energy. We see overall the reaction Gibbs energy is less than 0. So that tells us overall this is an exergonic reaction. Okay. Let's now talk about kinetic and thermodynamic products. So reactions can produce different products depending on the temperature, activation energy for each pathway, reaction enthalpy, and reaction entropy. The kinetic product is the product generated from the faster reaction pathways with the lower activation energy. The thermodynamic product, on the other hand, is the lower energy product. So for example, if we compare these two reactions, here is the activation energy for the kinetic pathway. So the pathway in green represents the kinetic product pathway because it has the lower activation energy. As we can see how the pathway in red has a higher activation energy than the pathway in green. However, the pathway in red generates the thermodynamic product. Why? Overall, the thermodynamic product is lower in energy than the kinetic product. It is important to note that the kinetic and thermodynamic product can be the same product in many cases. So it's when you're defining and classifying something as the kinetic or thermodynamic product, you first check activation energies. The kinetic product is the product made from the faster pathway with the lowest activation energy. The thermodynamic product is just the lowest energy product. Now, why is it important? Why do we care? Well, it's key to identify if a reaction is under kinetic or thermodynamic control. At higher temperatures, the activation barrier is more easily surmounted, and the kinetic product can undergo reversible reaction, followed by a forward reaction through the higher energy pathway to generate the thermodynamic product. The kinetic and thermodynamic product may be interconverted under equilibrating conditions. So, depending on the reaction conditions, especially the temperature, the reaction can be under thermodynamic or kinetic control. Generally, at lower temperatures, the reaction is under kinetic control, while at higher temperatures, when you have more energy to surmount this activation barrier and proceed through and initiate reverse reactions, so the two products can actually equilibrate with one another, then you'll be under thermodynamic control. So to practice identifying kinetic and thermodynamic products, I'd like you to tell me which products are the kinetic products and which products are the thermodynamic products for the following reaction. So pause the video, take a few moments, and then we'll discuss. Okay, so first and foremost, if we take a look at the activation energies here, I'm just going to draw this out. So as we can see very clearly, products E and F in this case are the kinetic products. Why? They're produced from the pathway with the lowest activation energy, and thus it is the faster pathway. The kinetic products are generated from the pathway with the lowest activation energy. Now we check overall energy. And as we see, products C and D are the lowest energy products. And thus, these would be our thermodynamic products. So it's really important to be able to read reaction energy diagrams and identify kinetic versus thermodynamic products. Okay, let's keep going now, and let's talk about the Hammond postulate, which is a vital idea to help us understand and talk a little bit about the general structure and behavior of the transition state. The structure of the transition state resembles the structure of the nearest species. What does that mean? Well, transition states for endergonic steps resemble products. Transition states for exergonic steps resemble reactants. For example, if we have an exergonic step where the net Gibbs energy for that step is less than 0, the transition state will be reactant-like. It resembles the species closest in energy to it, which are the reactants. By the same token, if you have an endergonic step, the transition state will be product-like. Why? The products are closest in energy to the transition state. What's so useful about the Hammond postulate? Well, the Hammond postulate allows us to estimate the energy and structure of the transition state. So let's take a look at the following reactions, and let's identify each reaction as exergonic or endergonic. And I'd like you to tell me, does the transition state resemble the reactants or products for each reaction step shown? So I'll do the first one, you'll do the second one. So here we have, for this overall step, if we take a look at this overall step, here's our transition state. The overall Gibbs energy of reaction is less than 0. This is an exergonic step. And as a result, as we can see, if we look at our closest energy species, the transition state resembles the reactants. And if we take a look at the transition state for this reaction, it looks very similar to our reactants. So with this example in mind, I'd like you to give attempt at the next problem. I'd like you to tell me, is this step exergonic or endergonic? And does the transition state for this step resemble the reactants or products? So pause the video, work through that, and then we'll discuss. So first and foremost, looking at the overall Gibbs energy for this step, it's greater than 0. This is an endergonic step. So looking at the closest energy species, the transition state resembles the products. And as you can see, if we look at the sketch for the proposed transition state for this reaction, it looks an awful lot like the product. So the Hammond postulate is incredibly useful. If you identify whether a reaction is exergonic or endergonic, specifically whether each reaction step is exergonic or endergonic, you can in turn determine whether the transition state resembles the reactants or products respectively. And this lets us then look at our reactants and products and talk about the transition state's relative stability and energy. Really powerful stuff. Now, as I've alluded to by the fact I kept saying step rather than overall reaction, many reactions are multi-step and go through what are known as intermediates. So many reactions occur in multiple steps, and these steps generate what are known as reaction intermediates. What is an intermediate? It is a species in a reaction that is neither the reactant nor the final product. One distinction that I have to make is that intermediates can be isolated, intercepted, and observed. The transition state, by its very definition, cannot be isolated. Okay, so for example, if we look at this multi-step reaction, we have 1, 2 steps, and in the middle, we generate an intermediate. Carbocations are pretty common intermediates. So this reaction, here is the intermediate generated from step 1 that reacts in step 2 to make the product. Intermediates are pretty useful to identify as they can tell us a lot about a reaction. Importantly, each step in a reaction has its own free energy of activation and its own transition state, and each step can be exergonic or endergonic. Now, thankfully, we can simplify our analysis when we look at multi-step reactions by considering the rate-limiting step. The rate-limiting step in a reaction is the slowest step with the largest activation energy. So if there are two possible pathways, the pathway with the lowest activation energy for the rate-limiting step will be the kinetic product pathway. For example, if we look at these two steps, 1 and 2, what is the rate-limiting step for this reaction? What is the step with the largest activation energy? As we can see, comparing activation energy step 1 versus activation energy step 2, step 1 is the rate-limiting step. And the reason why that's important is it allows us to focus our analysis on the rate-limiting step, because the rate-limiting step is ultimately what controls the reaction rate and what can often control the product selectivity. So by going through all of this analysis, by looking at all of these energy diagrams, we can in turn start to talk about what products are favored from a reaction, how favored are those products, how can we go about predicting the products of reactions in a systemic and efficient way. So, to showcase the power of this kind of reaction coordinate analysis, we're going to take a look at alkene hydrobromination, and we're going to apply the Hammond postulate. So again, let's keep in mind what the Hammond postulate says. The structure of the transition state resembles the structure of the nearest species. For endergonic steps, the transition state resembles the products. For exergonic steps, the transition state resembles the reactants. So we can use the structures of the reactants, products, and intermediates to estimate the structure and thus the energy of the transition state. So when we think about alkene hydrobromination, we've established, literally in the last example, that step one is limiting. It's our rate limiting step. So in step one, we take this alkene and we protonate it to make a carbocation. In step two, the carbocation gets attacked and we form a carbon-bromine bond. So we have established, looking at this energy diagram, that carbocation formation is the rate limiting step. Step one, where we make this carbocation, is rate limiting. Now, there are two possible pathways for this rate limiting step. The kinetic product pathway will have the lowest activation energy. This intermediate is a carbocation, and this overall step has a Gibbs energy greater than 0. This reaction step is endergonic. Now, what this tells us is if we take a look at our transition state, because it is endergonic overall, because this reaction step is endergonic overall, the transition state resembles the intermediate carbocation. Put another way, the transition state resembles the quote-unquote product carbocation from this step. Thus, if we think about this transition state for a moment, if I'm visualizing this transition state, it's going to be carbocation-like. So if we have two possible pathways, one where we generate a primary carbocation, and two where we generate a tertiary carbocation, the pathway forming the lowest energy carbocation will be faster. And if we had a choice between making a primary versus a tertiary carbocation, we are going to make the tertiary carbocation. So, because the pathway to make the tertiary carbocation makes a more stable carbocation, and the transition state resembles this product carbocation, this transition state is lower in energy, and thus the pathway to make this more stable carbocation is going to have a lower activation energy and will thus be faster. So this is why the Hammond postulate is so useful. We can take a step, identify if it's exergonic or endergonic, look at the reactants or products, and in turn, because in this case the transition state resembles the products, we can use stability trends for carbocation products to estimate the relative energy and stability for the transition state. Because a tertiary carbocation is more stable and lower in energy, the transition state to reach that tertiary carbocation will be more stable and lower in energy because they resemble one another. And in turn, the pathway to make the tertiary carbocation will have a lower activation energy and will thus be faster. It's amazing how much information we are able to derive just using simple stability principles and mapping out the reaction coordinate. So this explains why, if we look at our overall reaction diagram, you only see the tertiary carbocation pathway drawn, because this pathway is the pathway with the lowest activation energy. It is both the kinetic and the thermodynamic pathway. How do I know it's both the kinetic and thermodynamic pathway? Well, oh, here we go. Sorry, I circled the wrong one, this one, because it's making the tertiary carbocation. The pathway that makes the tertiary carbocation is going to be both the kinetic and thermodynamic pathway. It's the faster pathway, and it makes the more stable product. What we're now going to do towards the end of this chapter is we're going to do a survey of some common organic reaction mechanisms. We're going to talk about some common steps that you'll often have to draw and use in drawing organic reactions. So let's talk about the first case, which are addition reactions. So addition reactions, in essence, a pi bond is broken and a new sigma bond or sigma bonds are formed. In electrophilic addition in particular, a nucleophilic pi bond is used to form two new sigma bonds. To this end, the alkene is protonated by forming a carbon-hydrogen bond, and then the carbocation is attacked by the nucleophile. So for example, if we are drawing out the addition of hydrochloric acid to this alkene, we protonate the alkene to make a carbocation. For those curious, why did we make this carbocation? It is a tertiary carbocation, which is preferred over the secondary carbocation, as we observed from our prior case study. That carbocation is then attacked by the nucleophilic chloride ion, and that forms our desired carbon-chlorine bond. Just to make sure you're able to practice this skill set, I'd like you to, given these reactants, draw out a mechanism and draw out the products for this reaction. So pause the video, work through that, and we'll discuss momentarily. So first and foremost, we're going to protonate the alkene. From here, we generate a carbocation. The bromide anion then attacks the carbocation, and that forms the desired carbon-bromine bond. Now, in addition to polar mechanisms of addition, we also have radical addition, where a radical adds to a pi bond to form a new sigma bond. Radical termination then occurs. So for example, in this case, the bromine radical adds to this pi bond. This in turn generates a radical. Why at this position? It's the tertiary position which is favored over the secondary position. Tertiary radicals are more stable. From here, we abstract a hydrogen, and that continues on the radical chain. One thing I'd like you to note is that if you compare and contrast these two examples, if we compare polar versus radical addition, in radical addition, the halogen ends up at the less substituted position, while in electrophilic polar addition, the halogen ends up at the more substituted position. Just something to note for later. Just to make sure you're comfortable drawing out radical addition pathways, let's try drawing out the radical addition mechanism for the following reactants. So pause the video, work through that, and then we'll discuss. So first and foremost, the chlorine radical adds to the pi bond. That generates a new radical and a carbon-chlorine bond. From here, the radical abstracts a hydrogen, and that in turn forms our desired carbon-chlorine and carbon-hydrogen bond. The last type of addition is one that you'll see primarily in later chapters, but it's important to discuss it here, which is nucleophilic addition, where a nucleophile adds to an electrophilic pi bond to form a new sigma bond. So in this case, we have this carbon anion. We then attack the electrophilic carbonyl carbon. That breaks the pi bond and forms a new sigma bond. Let's practice drawing a nucleophilic addition mechanism. So here we have another carbon anion, and we have an electrophilic carbonyl carbon. So here, the carbon anion attacks the electrophilic carbonyl carbon, breaks the carbon-oxygen pi bond, and that in turn yields and forms our desired carbon-carbon bond. Wonderful. So we talked about addition. Let's talk about common reaction mechanisms for elimination. So in elimination, we're forming a pi bond by breaking one or more sigma bonds and losing a leaving group. One type of elimination is E1, where the loss of the leaving group occurs to form a cation, followed by elimination of H+ to form a pi bond. For example, in this case, our leaving group, chlorine, leaves to make a carbocation, and from there, a base pulls off a proton and facilitates elimination to form the desired pi bond. Let's look and let's practice drawing elimination mechanisms with this next example. So pause the video and try to draw the full E1 mechanism for the following reaction. Okay, so first and foremost, we lose our leaving group. That in turn generates a carbocation. From here, the base, be it water in this case or chloride, water is already floating around, so we might as well have it do the elimination. Pulls off a proton, facilitates elimination to form the pi bond. This in turn yields our elimination product along with water and H3O+. In contrast, another type of elimination that you see is the E2 elimination, which entails the concerted loss of the leaving group and abstraction of hydrogen by a base to generate an alkene. So in this case, the base pulls off a hydrogen. We, in turn, use those sigma bonding electrons to form a new pi bond, a new double bond, and we break the bond between carbon and our leaving group. This in turn generates the elimination product. So let's practice that, and let's draw out the elimination mechanism for the following reactant. This time, the base is tert butoxide. So take a few moments, and then we'll discuss. Okay, so here's our base. So as discussed for the E2, the base pulls off a proton. We use those sigma bond electrons to eliminate and form a new pi bond, and that breaks the carbon leaving group bond. That generates our desired alkene along with our leaving group. The last type of elimination is 1,2 elimination, which entails loss of the leaving group to reform a carbon-nitrogen or carbon-oxygen pi bond. Classic example occurs if you have a leaving group adjacent to a carbon-oxygen bond. The oxygen kicks its lone pair down, and you break the carbon leaving group bond. This reforms your carbon-oxygen pi bond and kicks out your leaving group. I'd like you to draw the arrows for this 1,2 elimination for this second reaction. So take a few moments, and then we'll discuss. So in this case, again, the oxygen is going to kick down its lone pair to reform the pi bond, and we're going to break this carbon-carbon bond to kick out our leaving group. You'll see both towards the tail end of this class and in 113, this example of 1,2 elimination where you end up breaking a carbon-carbon bond and kicking out a leaving group is going to be a pretty problematic reaction, as it often is an undesired side reaction in many reactions we discuss in 113. Okay, so we've talked about addition and elimination. Let's talk about some common mechanisms for substitution reactions. Now, what are substitution reactions? Substitution reactions involve the displacement of a leaving group by a nucleophile. In the SN1 reaction, you lose your leaving group, and then you get the attack of your nucleophile onto the resulting formed carbocation. So for example, in this SN1 reaction, we lose the leaving group. That makes a carbocation. And from here, the carbocation is attacked by your nucleophile, and that in turn forms a new bond between the nucleophile and the carbon. With that framework in mind, I'd like you to try to draw the SN1 mechanism for the following reaction. Remember, first you lose your leaving group, then your carbocation that's formed gets attacked. Okay, so first and foremost, we lose the leaving group. That in turn makes a carbocation. Let's draw this a little bit nicer. From here, the carbocation gets attacked by the nucleophile, and that forms a new bond between the nucleophile and our carbocation. For those wondering, what's the nucleophile? Well, we talked about that earlier on in this chapter. Generally, either an atom that's electron rich with a lone pair, or an atom with an electron rich pi bond. Let's talk about another type of substitution reaction, that is the SN2 reaction, which involves the concerted attack of the nucleophile and loss of the leaving group. So for example, the nucleophile attacks, and we kick out our leaving group. So this whole giant thing that we kicked out is the leaving group. So in effect, we formed a new bond between our nucleophile and carbon and broke the bond between carbon and our leaving group. Let's try to draw a mechanism for this SN2 reaction. This is actually a named reaction. It's known as the Finkelstein reaction. It's a pretty useful reaction, But in practice, it's a bit of a pain to run because it requires such a large excess of the nucleophile. So take a few moments, try to draw the SN2 reaction mechanism for this reaction, and then we'll discuss. So in this case, we have our nucleophile in the SN2 reaction. Our nucleophile attacks the carbon with the leaving group, and we lose the leaving group simultaneously. This is a concerted reaction. This yields our product and loss of the leaving group. Okay, let's talk about one other feature and one other common organic reaction mechanism, which is the proton transfer. In a proton transfer, a basic lone pair abstracts a proton from an acidic bond. The most basic lone pair is typically protonated first, and we can use pKa values to estimate the position and favorability of an acid-base equilibrium. For example, in this case, the basic nitrogen lone pair pulls off a proton, and in turn, we generate the conjugate base and conjugate acid. Considering that amines have a pK of around 35, while a ketone has a pKa of around 22, the delta pKa is about 22 minus 35, which is negative 13. So this is a product-favored pathway. One thing I'd like you to note is many reactions utilize intermediates that may be minor species at equilibrium. For example, protonation of a ketone is typically reactant favored. This protonated ketone species is highly acidic with a pK of somewhere around negative 3, while the hydronium ion has a pK of around negative 1.7. Which makes sense. This oxygen is sp2-like, this is oxygen's sp3-like, and in turn, if we take a look at the two bases, a lone pair on an sp2 atom is going to be more stable than a lone pair on an sp3 atom. Because this base is more stable, this protonated ketone is less stable, more acidic. One last thing, some other instructors, and I'm also very particular about this because it highlights a very important point, is this idea of proton transfers as intermolecular versus intramolecular. Intramolecular proton transfers are only possible when the acidic and basic atoms are close. For example, in this case, the nitrogen and oxygen atom are close to each other in three-dimensional space, and as such, proton transfer is reasonable to draw intramolecular. If you need to draw proton transfers in other cases where the acidic and basic groups are not close to one another and geometrically able to engage in proton transfer, it's best to draw intermolecular proton transfer. The way that you draw intermolecular proton transfer is by using the solvent, reactant, or product molecules. In this case, rather than drawing the proton just shuttling over to this bottom oxygen, instead, geometrically, that's not allowed. So instead, what's going to happen is that the solvent is going to facilitate this proton transfer. So we first pull off the proton from this oxygen atom, and then from here, your molecule then pulls off a proton from the protonated solvent. So the key thing I want you to keep in mind is that the solvent often facilitates proton transfers. So if you have a choice, unless it's really easy for the acidic and basic atoms to be close in 3D space, Just draw the proton transfer using the solvent or other molecules. Draw it inter rather than intramolecular. The last topic that we're going to discuss is we're going to introduce this idea of carbocation rearrangements. So in many cases, an intermediate carbocation can undergo a rearrangement to yield a more stable carbocation. So as we discussed, more substituted carbocations and resonance stabilized carbocations are especially stable. So you will often see rearrangements to generate a more stable carbocation. One type of rearrangement is a hydride shift, which is the shift of a carbon-hydrogen bond from an adjacent atom to the carbocation. So you're essentially shifting the carbon-hydrogen bond to the carbocation. So in this case, the carbon-hydrogen bond undergoes a hydride shift, and we get a new carbocation. Why does this occur? We went from a secondary to a tertiary carbocation, and that tertiary carbocation is more stable. By the same token, if you have the potential to make an allylic carbocation, you'll often see hydride shifts where the carbon-hydrogen bond shifts over. We make a new carbon-hydrogen bond and a new carbocation. And this carbocation is allylic, especially stable. You can also see what are known as alkyl shifts, which is the shift of a methyl or another alkyl group from an adjacent atom to the carbocation. For example, if you have a secondary carbocation, you can see an alkyl shift if it's possible to make a tertiary carbocation. With this in mind, I'd like you to draw an alkyl shift that would generate a more stable carbocation from the following compound. So pause the video, take a few moments, and then we'll discuss. Okay, so if we shift this methyl group over, after the alkyl shift, we have a pretty stable tertiary allylic carbocation. So again, alkyl and hydride shifts occur to make more stable carbocations. This is in part one of the reasons why carbocations can be a little problematic to work with, because they're often prone to these rearrangements and shifts. Now, forming a donor stabilized carbocation is also especially favored. For example, if you undergo an alkyl shift for this molecule, you see you generate a carbocation that's adjacent to a pi donor, such as this oxygen atom. This is one of the steps in a key rearrangement known as the pinnacle rearrangement. Ring expansion is also favored, so carbocations can also undergo alkyl and hydride shifts to relieve ring strain. So this cyclobutane, as we talked about previously, is very strained. And as a result, this cyclobutane can undergo an alkyl shift where the ring opens up and expands, and that in turn generates a cyclopentane, which is a less strained ring system. So just be really careful when you're dealing with carbocations because they can often undergo rearrangements and shifts, and it's important to be familiar with these mechanisms whenever you deal with carbocation forming mechanisms. So, as we finish up this chapter, we're going to continue on our lecture series with chapter 7. In future chapters, we're going to be delving into specific reaction mechanisms. So you're going to use a lot of the fundamental steps that we learned in this chapter, and you're going to apply these fundamental steps along with the process of analyzing reaction diagrams and reaction coordinates in order to understand in chapter 7, addition and some elimination reactions. And in later chapters, we're going to talk about substitution and elimination reactions in greater detail. So this concludes the chapter 6 lecture, and we'll be continuing on with the organic chemistry lecture series with chapter 7.